guess I just ask you to get your screens back on, and then we can have questions for all three speakers. And maybe while I wait for some more questions to come up, I can start with, there was a unanswered question to Morgan, unless you talked to Suzanne privately and answered it yet? No?
No, sorry, was I supposed to?
Oh no, I was just curious if you did. And where did it go?
Oh, I think I
here.
have it. The question, if you want to generate synthetic data, if we have confidence intervals for each parameter based on clinical data, do we need to do the simulated annealing again to optimize the virtual parameters? If you had, so usually what you get is clinical data of the output, so of the model prediction, as opposed to like something like a differentiation rate, or you know, you have some experimental data from that, but you don't necessarily have a clinical measurement of rates, which are what are usually we're using as to generate the virtual patients. You can definitely use, there are different strategies for generating virtual patients, and you can use priors like including confidence intervals that may be estimated from experimental data. You still generally need to do some culling step. So we found, for example, like if you're thinking about, unless it's very specific to the patient population you're interested in, so late stage non-resectable melanoma patients, you're going to end up generating patients that have tumor doublings that may be outside of the ranges that you're thinking of. So we use those clinical or those confidence intervals to inform where the sampling needs to be done. And you can either then look to see whether you have some other clinical predictor that seems to fall within a range that you want, or use a simulated annealing as described by Alan et al. To do it. So I think it's kind of maybe on both in the answer to that. I'm not sure if that actually answered the question at all.
Yeah, Morgan, thank you so much. Yeah, that's a very accurate answer. But just, you know, because for the COVID-19, and I have some models for the COVID-19 and also influenza. So then the parameters that they have estimated is from the patients, and they have one average value with the accuracy of 95%. So I was looking at your paper. So then you have looked at, and based on your presentation, you, in order to find the standard deviation, because you have generated the normal distribution. So based on this, so that standard deviation is coming from the literature, right? So then when I'm looking at this paper, so they have generated based on the 13 or 15 patients. So, and then they have the accuracy up to 95%. And I have generated, find the standard deviation. So when I'm generating the patients, the virtual patients. So then the parameters, for example, for the viral load is about the same for different patients. Mm-hmm. So then I was thinking not to use the simulated annealing, because what I'm thinking, you know, at your presentation, the standard deviation coming from the literature, right? So, and they have estimated. So then you have used the simulated annealing to make sure that it is between the physiological orders. Mm-hmm. But for my papers, they are coming directly from the patients. So I was thinking maybe, you know, the simulated annealing is something, you know, time consuming simulation. So I was thinking maybe, not to do that. So then, just
I think it depends, Suzanne, too, if you're only using one thing to generate. I mean, you always want to do a check to make sure that they seem there is some realism there. So I think you always want, even if you're just using a clinical like viral loads to generate the patients, you're going to want some sort of output that you're making sure that's not used in the generation step, to make sure that you're corresponding in terms of other perhaps like immunological parameters. But I think you definitely could do that. One thing that you might fall into is that if you use several parameter sets that are from different patients at the generation step, there's no guarantee that, you know, say you sample from a high part of the viral loads but a low part of the T cells, there's no guarantee that that matches in terms of what an actual patient in the clinic would have. So that's where that like validation step comes in handy, because you can get, we just finished a paper on this actually with Kathleen, where you can end up generating patients from wrong sides of the intervals and then getting really weird results. So.
Oh yeah. Okay, so then and I can share the paper with you and later maybe just to see the results and how. Thank you so much.
Yeah, no worries.
Thanks. I have lots of follow-up questions to that too, but I think Chapin, you had something you wanted to raise as well, right? I'll let you go ahead.
Oh yeah, I had a question for Katia about her bottleneck work. And so we're doing a bottleneck project as well on SARS-CoV-2, and we haven't worked with the MBA data, for example, that you showed, which was absolutely fascinating when I read that paper. But nonetheless, the viral load data that we are working with from the few different studies has fairly quick clearance. And what I've been thinking about, similar to what you had shown, at least qualitatively, and what I've been thinking a lot about lately is for long haulers and bottleneck, perhaps like a bottleneck event or the probability of transmitting non-neutral mutations in a long hauler case where the infectious period might be an order of magnitude longer than a typical case. And I'm wondering if you thought about sort of the dynamics of transmitting non-neutral mutations amongst long haulers as compared to a sort of atypical case where it's cleared on the order of maybe weeks.
Yeah, that's a great question. So when you say long hauler, you don't mean kind of the people who just have long-term symptoms, but actually have are chronic infections, basically.
Yeah.
Okay. Yeah. I'm very interested in that as well. So trying to learn from flu, another acute viral infectious disease. So there's been some really nice work out of, Tom Friedrich's group, as well as Adam Loring's group, et cetera, for looking at actually acute flu infections, and you don't see the evolutionary dynamics like within acute infections. It doesn't seem like there's enough time for like selection to really go on, that the dynamics are really, viral dynamics are basically driven by genetic drift, so there's not kind of, there doesn't seem to be, even in like, vaccinated breakthrough infections and so forth. And if someone's being treated with an antiviral, you still don't see immune escape, or antiviral escape kind of arise. So basically acute infections seem like it's just too short, for there to be any kind of, selection acting efficiently on the viral population. Now that's different, of course, in chronic infections. And so there's a nice paper by it was paper again by Catherine Schoen and Jesse Bloom, a different paper, where they looked at chronic infections in flu and E-life, and you do see, clonal interference, you see mutations arise, beneficial mutations arise, and so forth. And there's, and so that can definitely happen, I think, in longer term infections. And I think the same thing kind of holds for SARS-CoV-2. The papers I've read that I've looked at within host dynamics in acute infections, you don't see any indication of selection going on, but you except for like in experimental animal models and so forth, where you can basically you give a huge amount of virus and it's 50-50 D and G 614 or something like that, but you don't see kind of in natural infections, if you have such a small bottleneck, you have very little genetic diversity to begin with. You don't see de novo mutations arise and kind of fix over that period of acute infection. So for chronic infections, there's some really nice examples of showing that there's adaptive evolution going on over the course of kind of months, for sure. And there's speculation that for some of these variants, like the one in kind of in December in England, what was it? Well, Alpha, right? Whatever the name is now, that may have emerged in from kind of a chronically infected individual, with potential spillover, into the human population, and that's just because how divergent it was, from Wuhan, the Wuhan strain, and you wouldn't expect that to happen from just these series of acute infections. So I think tracking adaptive evolution in the viral population, chronically infected individuals, is really worthwhile. Whether they are, they contribute to kind of population level evolution or not. I mean, I think there's also things to be learned about, mutations that allow for immune escape and so forth, if there's enough time for that to happen.
Yeah, I've seen that paper as well. What was the name of the first author on that E-life paper with chronic infections and influenza?
It's XUE. It's called something like parallel evolution at multiple scales or whatever, because what they effectively find is in these four patients who are chronically infected, and I think it's because they've been immunosuppressed because of therapy, the cancer therapy, basically. The same, very similar mutations arise on the hemagglutinin protein, which is kind of like the spike, in flu, and they arise and reach high frequencies, and so it seems like there's still some residual immunity, that's driving immune escape. And then you do see those kind of pop up at the population level. So you see the same mutations at parallel scales.
Really interesting. Thank you so much.
Yeah.
James, you're mute. James, you're mute.
Sorry, I turned myself on mute so you wouldn't hear my dog. I'm going to attempt to see if I can get the discussion going to kind of bridge the in-host and the between-host a bit and think about immunity population level. I'm probably not going to do a very good job of it, but I'll try. But I was kind of curious, just starting out that, Freya, your transmission potential, and this is kind of a question for everyone, is that can that be considered a measure of population level immunity of some sort that takes into account ongoing restrictions or non-pharmaceutical interventions as well? Is that a reasonable way to look at that?
You could look at it as a, I guess, an expected reproduction number in the general population under restrictions, I think, under non-pharmaceutical interventions. As I said, I think it definitely should incorporate immunity, not just vaccine-derived immunity, but exposure-derived immunity. As we haven't got there yet, but I think we will. I think one of our challenges is going to be determine what that might be. So we're trying to work with our public health authorities at the moment to see if we can do some sort of prevalence surveys on a regular basis or similar to try and get a handle on that. I don't know if there's similar. I know that the UK has the ONS infection surveys, which has been providing them with that sort of information, but I don't know if there's a similar or equivalent in Canada. But that's if there is, I'm interested to know, because we can use it to demonstrate to our government that it's a useful tool.
Well, Jane's been working with seroprevalence.
Yeah, we've been working with seroprevalence. We have been discussing a lot, some sort of risk metric for infection based on different distributions of immunity. But that's as far as we've got. We have lots of ideas. We don't really have one thing that we think is going to work. And I've been talking a lot actually with Lauren, and I'm not sure if Lauren has a comment, but maybe she can also message in the chat too.
So what kind of what type of data do you use for those analyses?
So we try to calibrate our transmission model to seroprevalence surveys.
Oh, they are, okay, yeah.
But we have a large transmission model where we try to, estimate what the actual number of infections are based on age, and then we project what a what the distribution of immunity looks like, and then we try to figure out how that's related to protection and infection risk.
So to piggyback on some of what you were saying, Jane, I'm wondering what your opinion is about the waves of COVID that are observed. Whether and of course and it's in the popular media also, and it's we see these waves every 3 to 4 months, right? And it's a question about how much of it is based on immunity considerations versus changes in contact rates versus changes in the viral population. Of course, kind of here, at least in the in the US, the fall wave was Delta, right? So what's your opinion about how much immunity actually plays into feeding into these waves?
Well, we try to incorporate the changes in behavior into our model. So we incorporate public health mitigation into our model, and we try to incorporate some sort of compliance index and seasonality and changes in transmissibility into the model. So given all of that, then what we do is we project what we think seroprevalence looks like, though we have a lot of assumptions as well based on how long immunity is protective against infection and against severe disease, and we assume that those protective capacities are the same for all the different age groups. And we know that that's not necessarily true, with the waning rate being different in older individuals. But what we've actually seen really is that even the level of immunity that we have in the Canadian population right now is just not protective against infection, but certainly protective against overburdening the healthcare system in certain provinces, minus a couple who had severe relaxation in their public health mitigation strategies in the summer, and even went to the opposite effect in telling people they more or less should be relaxing their behavior back to pre-COVID times. So I think immunity plays a big role still in the infection risk. And in fact, what we've seen when we try to, we fit our model to data, and then we project and compare against other data. And we do see that a 3-year waning rate from full neutralization to full susceptibility is what best fits the outcomes. And so that means that you have a 1-year waning rate between like from a mild infection back down to susceptibility. So our model assumes that that's only 1 year protective for individuals. So I mean, it's all hypothetical, but that's what we're finding with our model.
But it is somewhat consistent actually with, if you look at seasonal coronaviruses, right, and the timing between infection and subsequent infection, right? There's that, I think it's Nature Medicine paper that looked at that, right? And it's on the order of about 1 year to 3 years, right, for reinfection. So it's definitely consistent with that.
Yeah, and that's why we were happy with that result. But and in fact, we started out only assuming that it was like that, and then we were asked, lengthen the period and take away waning immunity, and so we're able to replicate a lot of results that all these other models have. But it essentially just went back to model 1.0 what was best fitting.
And if you incorporate things like cellular immunity, like from original SARS, there's 10 plus years of cellular immunity. So what does that mean, I guess? So now you're susceptible to infection, but to go back to James's like between host, within host thing. So now you have what, like stepwise severity in terms of, because now you're being re-exposed and your T cells are reanimating. And so then, I mean, Rustam was talking about this as like a conversion to mild, children are exposed as when they're kids, and then it becomes endemic and not quite as serious. Do you think, does that jive with what you would think?
Yep.
Great. This
Yep.
is it.
Because we have a waning rate of protective, we have a waning rate that's related to neutralizing immunity, but also related to some average immunity where your probability of getting a mild infection increases. And so as you wane down, your waning rate from your immunity against severe disease is not waning at the same rate at which your immunity against infection wanes. But we could have like another level underneath, but we still need to like another waning rate, I guess, but we still need to like learn a lot more about T cells.
Yeah. Well, we'll help you with that.
Yeah.
So the time scale for chickenpox and the waning, isn't that for both infection and severe disease, isn't that quite a bit longer? Does that make a difference in those sort of conclusions about who you would protect? Or is it close enough together that it doesn't matter?
I don't know anything about chickenpox immunity. I know for pertussis, there's definitely waning. I don't know if anybody else knows anything about chickenpox.
I thought chickenpox was just was kind of sterilizing, so you can only get it kind of once. But I mean, the reason why you get shingles later on is because the virus stays with you for life. It just kind of hangs out latently. But I thought it was sterilizing immunity.
That could be. I might be confusing chickenpox and something
Yeah, which
else.
is why,
I thought there was a reinfection chance after. Yeah, but I guess it's.
I think it stays in your
shingles, yes.
spine. Varicella stays in your spine, I think, and then of course in times of stress, it comes back out as shingles.
That being said, I know someone that has had chickenpox 3 times.
Oh my gosh. Sorry.
So I think they just didn't get any immunity from their infections, I guess, but they had it symptomatic chickenpox thrice before they were 30.
Went to too many chickenpox parties, apparently.
Yeah. I'm wondering, Freya, if like when you incorporate infection into that, because I mean, you've opened your borders, right? So, and did you take away hotel quarantine?
In some places, yes. Yeah.
Yeah. So you will be importing cases.
Yeah.
And so I think it's going to be, in terms of like being able to observe infection in the population, given the level of immunity that you have already, I think it's going to be hard to figure out risk, isn't it? Because you're pushing people down to mild infections. You're going to have a lot of unobserved.
Yeah.
Yeah.
Yeah. We've been discussing ways to think about whether missed chains of transmission, and thinking about reasons for tests, whether you're a known exposure and unknown exposure and things like that. But we're pretty, I think we're a bit pessimistic that we'll be able to estimate the infection proportion from those methods without having some sort of like cross-sectional survey of people's level of immunity at some point in time, because like you said, we think that we know that case ascertainment is going to look very different in the highly vaccinated population to what we've experienced in the past. But also our population's also been very wired previously to get back to zero, and I think so testing rates have been very high, and that's been a big part of the response, and people's have been very like cooperative, I guess, with those public health directions. And so we've had very high rates of testing, and I think that with accepting a level of transmission in the population and accepting this sort of endemicity of COVID in the communities, I mean that people, I think our population's also just going to have a different approach to sort of getting a test, and we'll have we didn't have flu last year either, so we'll have sort of other viruses circulating with similar symptoms and things, and so so I think it's a lot of unknowns in so I think it would be a challenge to do it without having some seroprevalence, I think, surveys. And even then, I think it's still difficult to interpret and incorporate in our models as well. But I'd be interested if the work that you just described, is that published or in a technical report somewhere?
Yeah, it's posted on one of the archives, but if you email me, I can send it to you.
Okay, great. Yeah, I'd be really interested to have a look.
Okay, well, I think that now that it's 5:33, we can move on to the closing remarks. Yeah, I'm getting really hungry. So I'd like to thank all of the speakers from the conference and through the panels. And I'd like to thank everyone who supported the organization of the workshop, and James for moderating today, and Ed for leading one of the discussion panels as well. Thank you, everyone. I think that we need to think more about where we need to go with modeling immunity, and how to link the between host and the in-host components of immunity. And I look forward to having another small workshop in person sometime post-COVID. That would be great to have some small working groups really also thinking about these topics too. So thanks, everybody. Have a great evening / have a great tomorrow, Freya and James, and have a great time. Thanks, everybody.
Thank you very much. Bye.